Hello there.
Greetings, everyone. Welcome back to Underqualified. Thank you all for rejoining us. We got what I hope is going to be a really interesting episode today. Um, I am going to talk about the genealogical editing and biological research tool known as CRISPR, and Benjamin is going to branch out into sort of the general ethical complications that come with scientific research advancing faster than our moral understanding is advancing. So, lots of big things to talk about today. But just to get those of you caught up who might be new, there are 3 rules to this thing that we used to call a podcast, but I'm now calling a web show. The first is that we will do everything in our power to only say things that are true, but because we are underqualified, sometimes that doesn't always happen.
Quite often, if it's me.
The second is that if you are someone who knows more about whatever it is that we are talking about than we do, and you either just want to share in the discussion or you have something to add to the topic that we got wrong, you are required by international law to participate in the discussion and tell us what we got wrong, because this is all about sharing and learning new information. And finally, and most importantly, rule number 3 is be nice to us, because the show is called Underqualified, so we do kind of say from the outset that we don't necessarily know what we're talking about.
Sure don't.
But CRISPR is one of the coolest discoveries in the modern biomedical era. And I know that I say that a lot because I've talked about several really cool things that have been discovered recently, but I am standing by that claim for this topic. And proof of that is that Emmanuel Carpentier and Jennifer Doudna, D-O-U-D-N-A,
Nailed it.
won the 2020 Nobel Prize in Chemistry for their work on CRISPR-Cas9. So the community at large thinks that this is a big deal as well, not just me. So the first thing is that CRISPR is technically a process, which is something that I think gets kind of misconstrued, in particular just in my own experience, because I thought that it was like a thing. You know, you could like plug something into the CRISPR machine.
Yeah.
You know, but it stands for clustered, regularly interspaced, short palindromic repeats. That's a mouthful. So it's spelled CRISPR if you're interested in doing some research on your own. And actually what it is an immune system of bacteria, which I had no idea. It was originally discovered in Archaea, which is just a different type of prokaryote, and then again in bacterial species by a researcher in Spain. I can't remember the name of the university. I apologize. And so essentially what it is, we've talked about the immune system of humans actually a good deal in some earlier episodes of this. And so these repeated sequences, in between the sequences, are sections of bacteriophages that had attempted to infect the prokaryote.
Okay.
And so it works very similarly to the way that our immune system does, in that it's an adaptive immune response. So it remembers these interactions with the other species. But where it came from is not anywhere near as important as what it can do. And because of the way that it has evolved in these bacteria over the millennia, is that the CRISPR-Cas system, you'll sometimes hear it referred to as CRISPR-Cas9. The Cas9 portion is just CRISPR-associated spacer proteins. That's the Cas. Is just an enzyme associated with these repeated sequences, can cut very specifically in a genome when provided a genetic sequence. So a brief video that I watched talked about giving a genetic sequence to Cas9, which of course you would like inject it into the system. There's primers that you can actually buy if you're doing biological genetic research. There's many people who have created a lot of these sequences already, and you can just sort of like plug them into your system.
Yeah.
And it can very specifically go in vitro into the DNA and make cuts, and then you can actually reinsert new genetic material into that space that you have just bisected out of the genetic structure.
Sure.
And so I have an article here from the broadinstitute.org that's talking that this brief video is Dr. Feng Zhang, I'm going to say is how you say his name. And he gives a really basic example talking about typos in writing. And so he uses twinkle twinkle little star. And so you're reading along and it says twinkle twinkle big star, which of course is incorrect. That's not how it's supposed to go. And so there's a couple different ways that you can go about that. You can just remove big entirely. So you would just have twinkle twinkle star. And this is, by the way, is a complete ripoff of this video that I just watched. This is Perfect. None of this is something that I came up with.
Snatched.
Or you could simply add in little. So you'd have twinkle twinkle big little star. Well, that's not really correct either. And so it sounds sort of silly to talk about it this way, but it becomes a really complicated and important process to do all of those steps in the correct order when you're working with genes. Mm-hmm. So you need to go in, you need to bisect out the quote-unquote big portion of your genetic sentence that is incorrect, and you need to reinsert the little appropriate genes that you want into that area. Right. And so it starts out as a pretty simple concept and then gets vastly more complicated as you start talking about it from a biochemical standpoint. But it's an extremely powerful tool because when you initially talk about it, they say, well, you can give Cas9 a genetic sequence and it will go into the genetic system and find it and cut it out. You can actually give it like tens of thousands of sequences at the same time, and it can go in and it can make cuts in all of these places. And then you can go back in with RNA and just recode as much of that as you need to. So the applications for downstream research and biomedical uses are immense because of how expansive the capability is. Mm-hmm. But already, CRISPR is outdated.
Wow.
Which is really funny to say. So CRISPR or CRISPR-Cas9 is sort of the original process. And we have since... researchers are continuing to use CRISPR-Cas9. I'm not trying to say that it's not done anymore, but we, as if I had anything to do with this, scientists have come up with a CRISPR CPF1 system, which is just a different enzyme structure to the Cas9, that is less complex and more accurate than CRISPR. Because obviously... or than the Cas9 enzyme system, I should say. So the Cas9 complex requires 2 RNAs to function, and the CPF1 system only requires a single RNA, so it's smaller, it's easier to deliver into cells, and it cuts DNA differently than the Cas9 system does. So when the Cas9 complex comes along and cuts the DNA, it cuts both strands in the same place. So if you're thinking about your double-stranded DNA and they're running together like this, the Cas9 will come along and just like a cleaver, it'll just chop right through the genetics on both sides and it'll pull out that section. So that sounds really nice and clean, but if you've taken a genetics course, you know that that is actually really bad because having ends that bisect right on the same spot is just begging for mutations. Because it's a really unstable area right there while you're waiting for the RNA to come back in and recode your section.
Right.
And so that's called, it's cutting blunt ends, so to speak. And that leads to mutations as they're put back together. The CPF1 complex cuts the strands in an offset. So it kind of goes like this. If you're like viewing the genetic code left to right this way, it kind of goes burp, burp, burp. And so you have one strand that ends up being longer than the other. And while it sounds like that would probably be something that you don't want, that actually helps to stabilize the segment until you come in and reinsert the genetic code that you're looking for. And so that gives you not only the ability to remove portions of the genome, but then to reinsert the ones that you're looking for. I mean, it would be an impressive tool if it just cut stuff out of the genome.
Right.
But the ability to then go back in and essentially recode is extremely valuable.
So you said that the new technology is more accurate than CRISPR was.
Yeah.
What happens when CRISPR doesn't hit the target?
So the issue comes into play. It's just called, I think it's called off-target splicing. We very briefly did a quote-unquote CRISPR lab my senior year in college, and ours did not work very well. We were attempting to cut what's called GFP, or a green fluorescent protein, out of a bacterial model. So when we started, our cultures glowed green, and if the CRISPR process worked appropriately, when we were done, they shouldn't fluoresce anymore. And so what we ran into is that you can have off targets, and this is why I would say that the medical portion, of course, the Nobel Prize of 2020 was just awarded, so to say that it hasn't happened yet is very presumptuous. This is right on the cutting edge of biomedical science, but it has not happened yet because so much of our genetic structure is similar. It's all just A's, C's, T's, and G's. You're going to have areas that are similar to each other.
Yeah.
If you reach a level of similarity that's above, I forget what the exact percentage is, but let's say 75 to 80%, the enzymes that you have provided and the RNA sequences that you've provided to the Cas9, there's chance that it'll see that and it'll be a close enough match that it'll bisect it. And that's really dangerous because first of all, you probably won't know where it is in the genetic code because you weren't prepared for it to do that, and who knows how it will be put back together. And so that is a huge hurdle that this is going to have to get over in order to truly become a medical tool as opposed to a scientific tool. Because we used it in lab and we said, oh, well, ours didn't work, we didn't get GFP out. Bummer. It's not that big a deal. But if you're using this in human patients and that happens and you start cutting DNA in half in places that you're not aware of, you're going to be creating so many more problems than the one that you're attempting to fix, that really sort of removes this from the ability to be used. But the more accurate that it gets with the CPF1 and whoever, whatever's going to come after that, because there's always the next best thing in the works, so whatever system that is the next system is going to be even more accurate and more simple than the previous one. You might reach a point where you could go in and have a CRISPR treatment done, which is where the sort of the awe in the scientific community for CRISPR comes from, because the level of understanding of genetic diseases and cancers is ever expanding, but there's still so much that we don't understand, because genetics are incredibly complicated and every type of cancer is a different mutation. We always just talk about cancer as sort of an umbrella term, but it can mean so many different things. And CRISPR is one of the things that is on the precipice of actually addressing the problem. So what I mean by that is that if you have something, let's say you have cystic fibrosis, and I've talked about cystic fibrosis before because it's a genetic condition that I understand with a little bit of fluency, and is also, in some people, can be pretty straightforward. It's a misfunctioning or missing protein in one portion of the lungs that allows fluid to accumulate. So the treatments that we have now remove the excess fluid. So they treat the symptom that you are experiencing, which is that you're getting excess fluid build up in your lungs that shouldn't be there because your system's not working the way that it should be. CRISPR could go in, theoretically, hypothetically, cut out the portion of your genes that code for the protein that is currently not working, and insert the genetic sequence to get all of your cells in your lungs to express the appropriately designed protein, and your cystic fibrosis would just go away.
Mm.
Which, I mean, you can see why you would award a Nobel Prize for that. I mean, that's absolutely earth-shattering capability.
Yeah.
I mean, and, you know, there's mutations in, well, all cancer comes from mutations of some sort or another, but some of them, and I do want to do like a series on the way that cancer works. But you talk about the RAS protein cascade a lot when it comes to cancer, because that's a really common way that cancer happens, is you get a mutation in that string of proteins, because that's what leads to cell division, obviously. So if you break that, then the cell division is going to continue. And so again, you could take a patient, hypothetically, who has cells in their body that have hyper-functioning RAS protein systems, and just go in and take it out.
Right.
And there wouldn't be chemotherapy. You would probably need surgery if there's a substantial mass to remove it from the body.
Uh-huh.
But if there's no genes that are malfunctioning, there was no cancer. And so the impact that this could have on the medical world is immense, because we don't have, what I'm getting at is, we really don't have a treatment for the vast majority of genetic diseases or cancer at this stage in the game. Because what we do with cancer is we try to just kill the cells that are broken.
Uh-huh.
We remove them via surgery, and then you take the really nasty chemotherapy drugs to hopefully kill those cells so that they stop replicating. But that's not really fixing the problem. It's removing the problem. Mm. You're just, you expunge it from the body via surgery and using these medications. But you could just get all of the cells to work again.
Talking about cancer, or talking about...
Well, so you could fix... Yeah, either way, you could, there wouldn't be a need for medication, because it's just a procedure that you would go in and have done, and you would just fix all of the cells.
So if for CRISPR with cancer, again, it's all hypothetical because there are no clinical trials at this point.
Not even close. I want to make that very clear, that this is not something that's happening right now.
But is the idea that you're using CRISPR to treat the damaged cancerous cells, or that you're using CRISPR to inform your body's immune cells so that they attack the cancer?
You would target...
I guess I'm asking, is it for cell therapy, or like, are you just cutting out the damaged cells?
It would be for cell therapy. So hypothetically, you could go in the cells that are currently replicating far too rapidly and are creating that cancerous mass. You could target them with the CRISPR procedure and just remove and replace the section of their genetic code that is causing them to replicate too quickly. So there are several caveats to that. The first is that CRISPR-Cas9 system itself, like I said, there's accuracy issues. And so we're a long way, before we even begin to talk about the ethical considerations, we are a long way from even being able to test it from a physical standpoint, because there's no way that we're going to start testing this on higher, I hate to say higher living beings, but you know what I mean, more advanced systems of biological life or humans, until we can get it to only work where it's supposed to. Because if you go in to try and remove a portion of a cell that is creating a cancerous mass, and you just dice it up in a bunch of places that you're not supposed to, and all of those places mutate, you've just compounded your problem. But it's a really clean, it's a really clean system, in the sense that it itself is capable of cutting DNA strands on its own. So you don't need to like hijack the body. You don't need to bring in, the RNA transcriptase that the body already has. like self-contained. It can do this all on its own. You don't have to pair it with any like cleaving enzymes or anything. I'm taking this from the Broad Institute piece. And it talks about how tens of thousands of RNA sequences have already been created for the scientific community. You can just order them online, and you can use them in your research. And so the speed at which it occurs is really what has set it apart from the previous gene editing technology, because there was types of gene editing that you could do in a lab prior to CRISPR, but because of the ability to hold all of those RNA sequences at the same time, the capability is exponentially higher than those previous ones, and it's actually now being used as a diagnostic tool in the laboratory setting, because if you can just tell it to look for these genetic systems and put it in to your test tube or your cell plate, it'll run and find if and where that specific code is anywhere in your sample. And so you can use it to actually test for things like genetic diseases, because you say, well, we know that if you're going to have this condition and this type of this condition, you're going to have, ACCTGG, blah, 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 specifically. And so we're not going to have to do a ton of blood work. There's not going to be all these scans. We're just going to take a brief blood sample. We're going to take it into the lab. We'll run a CRISPR process, and it'll say, bam, there's one here and here and here and here and here. And it's really clear that you have that mutation. So this guy, Feng Zhang, and his team have helped create and share, this is the very end of this article, over 40,000 CRISPR components with academic laboratories around the world.
Wow.
So this is a process that has absolutely exploded in the biomedical and genetic and biological in general research community, which is fantastic. Because as someone who thinks that, cutting edge discoveries in biological research are really cool, it's really great that they have this rapidly created this much material to allow other people to work on it. Because the more that you're willing and capable to share with the scientific community, the faster things go. And obviously, the specifics of this science are far above my pay grade. That's why I was spending a little bit more time looking at the article than I have been in previous talks, because it is really powerful and it is really profound, but I don't want to be saying things that are inaccurate. And so there's really impressive and really exciting things on the horizon if the community can start to pin down, accuracy, getting it to cut, first of all, always where you need it to and only where you need it to.
Right.
So it has to first identify that section in your genetic code, and then it needs to cut it out every time. If you're only getting a 50% fix, then the problem's probably going to persist. But people are working hard and fast on figuring this stuff out, which trends us in the direction of your problem that you've brought to the table to talk about today, in that in the last 100 to 150 years or so, there have been instances of discovery like this that thrusts humanity's capabilities, let's say, a long way out in front of our legislative and moral understanding. And so there's some pretty serious negative connotations and possibilities with CRISPR specifically, of like you could go in and fix the genetics of a healthy individual who doesn't need this procedure to express genes that you want them to express, which is profound in a medical setting and extremely concerning in an ethical and racial understanding. If you're just getting this because you're wealthy and you want to be a few inches taller, so you go in and cut some genes out and reinsert some things. The abuse of this power, of course, is not something that we've gotten to yet, but is something that is concerning and worth talking about in the future.
Definitely. And I think that one of the phrases that I noted that you just sort of mentioned in passing was, before we even get to the ethical stuff, we have to recognize that we don't have the means to practically apply this technology yet.
Absolutely.
And that in and of itself is sort of the problem, because we don't feel the need to address the ethical concerns until the technology exists, which
that's an interesting point.
Which inherently is why we fall behind. Because if there's no framework, and of course it would be difficult to design for technology that is
we don't have yet,
hypothetical, but I'm just pointing that out, as that's why we don't keep up with our ethical standards for technological advancements, because we can't design the legislative structures to control them until the technology exists. So the way to get out in front of it would to be setting up regulations and ethical standards for CRISPR now, even before it's been being used in clinical trials. And that doesn't just apply to CRISPR, that's everything. But
that's just, that's a practical nightmare to contemplate realistically, because you don't even know what form it's going to take.
You don't know what it can be used for. There's the stuff that we think it can be used for, but it's not actually doing anything. And so really, I guess it would be more helpful to be generally having a conversation about genome modification and the adjustments that we can make to people's genetic codes. And that's maybe not on a legislative level, but on a cultural level has been a discussion for quite some time, because a lot of the technology that we're talking about still feels to me like science fiction type stuff, and probably to your average person is very, very much feels like the technology of Star Trek, or
absolutely,
or maybe more appropriate, like Brave New World or something like that, where it's a dystopia where things have gone wrong,
started promising,
exactly, in the efforts to better humanity, and then it is derailed. And so one of the best examples, or older examples of ethics in putting a stop on scientific advancement is talking about cloning, which that technology, well, the concept is ancient. It's
and there's proof of concept. I think that is something that a lot of people don't know, is that we have cloned
a number of
biological
animals,
organisms.
It can be done.
And a while ago, I mean, there was a lamb in
Dolly. I have that article pulled up. It's just very long. Dolly was born on the 5th of July of 96, so that's 25 years ago. And it was interesting because, and again, it's sort of the reactionary effect of once we have the technology, the ethical concerns sort of slam down hard, because Dolly, once the news broke about Dolly in 97, totally unprecedented, and just in the next year, Argentina, Austria, Brazil, Denmark, Georgia, Germany, Iceland, Mexico, Norway, Peru, Slovakia, Spain, South Africa, and the United Kingdom all banned cloning.
Didn't take long.
And in a few years' time, at least 30 countries had followed suit. This is a academic British Medical Bulletin article that I'm pulling from. And so it's just interesting to see how that played out, because it says the nuclear transfer for a non-human mammal is a fairly innocuous procedure. And that didn't really raise very many ethical concerns. And following Dolly, scientists cloned cows, mice, rats, goats, pigs, rabbits, cats, horses, and dogs by similar methods without really much pushback from anybody. And so and additionally, researchers have cloned human beings by embryo splitting to aid with artificial reproduction, which is
the IVF and
right,
stuff like that.
right, which is sanctioned by the ethics committee of the American Society for Reproductive Medicine. So that's all, it all checks out.
Because if you can get over the cost hurdle, it's incredibly expensive.
Right. But more embryos in the womb increases the chance of a successful pregnancy.
Right. Absolutely.
So, that's the thought process, and it's condoned by an ethical authority. And so the problem's not inherently with the reproduction of human beings, but the ethical issue seems to exist with human beings being produced by nuclear transfer, the cloning of human beings, basically, making identical copies of a person.
Yeah, I was going to say that I think that it's not a concern with infants because that individual, the individual that infant would become, or not even infants, embryos, excuse me, doesn't exist yet.
Right.
And so the issue comes into, like, when I say, I want another me.
Right. And so there have been arguments on an ethical level that cloning violates human uniqueness. It's a violation of dignity, and a variety of other things. Basically, the idea, and this is sort of a loaded word, but that it's unnatural.
Yeah. And get into playing God.
Yeah, and we're going to talk a lot more about that with some other concerns for sure.
It's a thing with CRISPR as well.
And the other thing that was interesting about just the smackdown from so many countries putting an outright ban on this is that, so a ban is different from a moratorium, right? Because a moratorium is basically just a temporary period of not researching. in order to there's not really like a set endpoint for that, but it's a temporary period. And it was just an outright ban saying,
nope, never again.
We're not doing this ever again. We don't want to know anything else about it. We want it to stop here. And so, it's always sort of the give and take of the desire for progress, both in a practical sense, how could we apply the new technology to better humanity, and also just, of course, the pursuit of scientific knowledge for the knowledge itself, for figuring out, is this possible, can we do it. There are lots of things that that money goes towards for research that don't really have practical applications, but because we're human beings and we like to see if we can do stuff, we still do it. And then there's the pushback of, well, maybe we can do this, but really, should we be doing this? And
the Jurassic Park
sort
of situation.
Yeah, we can bring the dinosaurs back.
We didn't talk about whether or not we should.
But should we do that? And they keep doing it for like 5 movies.
Several movies, yes.
It's a real head shaker. So the most contested area with cloning being making babies who grow up to be fully grown adults and members of society who are clones. And so that is human reproduction is at the heart of the issue of the ethical concern. And there are lots of potential hurdles of, like, is this a Star Wars type situation where now if I'm efficient enough at my clone production that I can just print an army and throw it out there and it's totally expendable.
Well, and like a Captain America type army, like you
or can I use the cloning technology to not only make clones but pair that with genetic enhancement to make like super soldiers. So that's very science fiction, but it's what comes up in popular media. And to be honest, it's not entirely far-fetched based off of the technology that we have.
No, I mean that's I won't say feasible, but considerable, if that makes sense.
Right.
Like you could talk about it in practical terms.
Right. Of course. Yeah. And so, it's sort of generally what I was referring to before, that the technology doesn't go away, and therefore the ethical consideration doesn't go away. And it's hard to say how long just everybody saying we're not going to do it is going to hold up for. And so it seems like there's still conversation there to be had on, if this is going to happen, what are the regulations for it, and how can we come to a consensus on that, because currently there is no consensus. So I'm going to move on from cloning a little bit, get into some other fun ethical discussions, and also talk a little bit about the ethical conversation in pop culture. Specifically, there's a 1997 film directed by or produced by Danny DeVito, which I did not know.
Really?
Yeah, but he knows what the movie I'm talking about. It's called Gattaca.
It's a really good movie, honestly.
Achieved critical acclaim.
Yeah.
It was a box office flop. It had a budget of $36 million and a box office total revenue of $12.5 million.
Ooh.
So that's a big swing and a miss. But
I would recommend it as a nerd. I have watched it. I thought it was quite good.
I haven't watched it in its entirety, but we did watch scenes from the movie in my 7th grade science class. So shout out to Mr. Plimpton at West Middle School.
Yeah.
But so basically the idea in this film is set in the future, and eugenics is widely practiced.
The way.
It's what everybody does. And eugenics is just the idea of, I guess, encouraging the reproduction of those with favorable traits and discouraging the reproduction of those with negative traits. And it was a whole big movement in the early 20th century, and it's not so good.
Nazi Germany
led to lots of not so good things.
Yeah.
And excuse me. And so there are just lots of little interesting tidbits to talk about in the Wikipedia breakdown of the plot of the movie.
Okay.
And so one particular thing is genetic discrimination is outlawed in this dystopian future, but practiced genotype profiling is used to identify those who have had their genes altered and specified, which is called being a valid, I guess, in the movie. And if you were conceived by natural human reproduction methods, then you're invalid. And
it's pretty cut and dry.
Yeah. And so your genotype is used to qualify you for employment, and invalids, those who were born through traditional methods of reproduction, are relegated to menial jobs. So technically illegal, but of course
happens everywhere.
Yeah, just because it's illegal doesn't mean that it doesn't happen. Yeah. And so basically there are impacts on the main character is Vincent, and he was born. He was a natural birth. He was traditionally conceived. And,
That's a very PG way of putting that.
Thank you. And his brother Anton was created through genetic selection, and he has all the good traits that their parents had to offer. And Vincent, on the other hand, is his genetic profile. He has a high probability of disorders and is only estimated to live until he's 30. And so basically Anton is like this super child, and Vincent is constantly losing to his younger brother and not being able to perform at a high enough level. And so and because he's very aware of what's in his genetic code, it's really difficult on him mentally and emotionally to know that he's basically from the beginning not supposed to amount to anything.
Yeah, he is the lesser of the two.
Exactly. And so I won't spoil everything about the plot in case you want to go watch this film. It's not a movie review. We're just talking about some of the points that it levies. And so there's another character, Jerome, who was a valid, so he was created by genetic modification, and he was a professional swimmer. And he this will be a little bit of a spoiler, so skip ahead by about 30 seconds. But he loses his ability. He's paralyzed from the waist down. Yeah. And this is revealed later in the movie to be because he threw himself in front of a car after getting second place in a swim meet. And so basically, the the impact on the other end for those who have been genetically modified, who do have all these positive traits, are from the very beginning designed to be successes. Is that when you fall short of that, right, it's more difficult. You are failing to be what you were designed to do. And so that's a big issue as well. And so basically, without giving away too much of the plot, Vincent is able to achieve much more in this society than his genetic code would have said he should be able to.
He was like a researcher or something, right?
He basically fakes his way into going to space and working for a space company.
That's why. I remember.
Yeah. And so he's showing himself to be intelligent, capable, competent. He's saves his brother's life a couple of times in this movie, and so that's a big thing. He's not supposed to be able to do that,
right?
Because Anton's this superhuman dude. And so that's, I think, this one of the central messages of this film in pop culture is, just because your genetics say one thing, but they can't be used to determine your worth as a human being. And those, Vincent was at risk for a lot of these disorders, and so it's saying, being at risk for those or having a disorder doesn't make you less human, and it doesn't reduce what you can offer as a person.
Well, and a risk is not the same thing as having a disorder,
right? Yeah.
Which, because there are parts of Gattaca that can happen today, which I think is something that might not be super common knowledge, but you can be, I made a joke about the 23andMe, but you can do that and look at what genetic diseases, if any, you are at an elevated risk for based on your genetic code. And so that's, it's not the exact same process, but that sounds like something that is, directly out of that movie.
Yeah, exactly. And so, I think that the film it's still science fiction, obviously, but it culturally sort of crystallized a lot of the discussion about genetic modification, and so geneticists, it's important culturally just to understand the perception of the science by society at large. And the science is continuing to advance, and thusly we're hurtling rapidly towards these very ethical concerns that up to this point have only been expressed in media, and it's terrifying enough to think about just in media. And so,
because you really could follow the premise of Gattaca with the technology that we currently have, because if I remember correctly, they're just choosing an embryo based on the genetic recombination of the parents. So it's not like they're inserting genetics, you're just picking the best offspring that you could possibly have as a combination of your two genomes,
right?
Which is something that is not done. It's not done, but you could.
Right.
That's something that we could look at today.
Right. And I think just to underscore the importance, Gattaca, it depicts a society that is overwhelmingly transformed by the development of new technology. And you might say, well, would it really be that much of a change? And I don't have the answer to that, certainly, if we're talking about what do I think is going to happen if we have technology like it's depicted in that film. But I think, you look no further than these things right here, smartphone, to realize how vastly and how rapidly new technology can alter the way that our society functions. And so the idea that it's not going to rock the boat, it's not going to need to be addressed by the structure of our society, that it's not going to need to be legislated on, I think is silly, because we're talking about, in the hypothetical, we're talking about literally determining the path of somebody's life in some ways,
before they even exist,
before they're even born. And you think about the vast number of discussions that the internet has generated, and that's in many ways less world shaking of a development than being able to say this kid is going to pop out with blonde hair, blue eyes, and is going to be able to run a 4-minute mile.
Grow faster, be smarter, perfect eyesight.
Exactly. And so there are tons of ethical concerns with web privacy and with how we manage our information and with how data is shared and collected. And certainly the structuring of society was not prepared for all of those ethical concerns to be levied. And so again, that's coming back to this idea that if we are constantly reactive to new technology being developed, we're always going to be behind the ball.
Just think about how the definition of privacy has changed over the last 30 years or so.
Yeah, definitely. And I think that's, going forward into this century, certainly going to continue to be one of the defining factors of the generation, is how is our data handled online? Is it a good thing that we can be marketed to more effectively based off of our Google search histories and
words that we say while your Amazon Fire Stick is in the room?
Right. Is it do we just say, oh, that's great because as a consumer I can buy these things that I actually want, or is that terrifying because we are essentially being documented by an algorithm?
I can personally say that I don't like the Amazon Alexa and the Google Home. Mm-hmm. It weirds me out.
Mm-hmm. I know some people don't even like the function on Gmail where it will offer a suggestion for what the sentence that you're trying to make is, or you'll type out a little bit and you'll be like hello, and then it'll throw the person's name in there, because it's like this computer, it knows how I work,
send emails on my behalf.
And so there's just so many unknowns, and the technology is still so nebulously defined. We're talking about ethical considerations for stuff that isn't happening,
doesn't even exist.
But that doesn't mean that we shouldn't talk about it, that we shouldn't continue to have structures in place to monitor these things. And there's this school of thought that has been used with that was used with cloning, that's used with other advancements, to say that advancements in technology, scientific discoveries are always connotatively like neutral. They're neither good nor bad. And that sort of recuses the researcher from responsibility for the impact that their discoveries can have, which is great for science on the level of pursuing ideas out of scientific curiosity. And a lot of times, science gets attacked, perhaps needlessly, for, what we were talking about being unnatural. That school of thinking, and that's not good either. But I still am wary of completely disregarding this idea that we should be formulating hypotheses of how new discoveries can impact society, what it could lead to, etc., and making informed choices based off of that. Because I pulled up this ethical, like this newscientist.com, talking about some of the biggest ethical considerations that are on the horizon, and you have to pay to read any of the articles, and so no. But it was just interesting to flip through some of these that are like, yeah, these are things that people are talking about right now. And it's, should we edit our children's genomes? Should we make guide society towards a world where everyone is thinking similarly, because societies where with like-minded people are often happier and safer, but what is the cost of that? Should we abandon privacy online? Should we give robots the right to kill? Should we let synthetic life forms loose?
I should just like bookmark this, and we can just do episodes on all of these.
Should we geoengineer the planet? Should we impose population controls? That's totally taboo.
That's crazy.
Should we colonize other planets? Should we stop doing science? Just
be like, we're good here. This is
scientific. It says scientific research may lead to benefits and advances, but they seem to go hand in hand with death and destruction. That's a little bit on the nose, but it's still something that people are talking about and concerned about. So I essentially offer no answers on any questions that are levied by scientific advancement. I'm just saying we should talk about them.
And I will say that the philosophy of the pursuit of knowledge for knowledge's sake is a really valid one, and it is one that a lot of people feel. And I would say that I feel in a lot of aspects that just understanding the world is a good enough reason to research something. But that is not the way that funded research works anymore. And so I just want to be very clear, don't misconstrue that, like, if you get a job as a college researcher or an independent researcher, you can just go into your lab and do whatever you want. That is not at all the case. There is oversight for everything. There is approvals and meetings and petitions on your part to the higher-ups of whoever it is that you work for every part of your research. Which is why research with, I'm going to say higher organisms again, is often so slow in occurring, because the hoops that you have to jump through, rightfully so, are extensive. I had a professor in college who did research on cows, and I won't divulge any of it because, of course, it's not published, but he worked with living cows and with their tissue. And I remember him talking about the paperwork that he has to fill out for the rights to do that research. And so there are things in place to keep scientists from just like wandering in a back room and doing stuff.
Right.
But yeah, there are definitely, we've reached a point where some things are beyond the discussions that we've had in, like Congress or something like that.
Right, and these are one of the harder things about it, and this was in one of the scholarly articles that I had pulled up, was it's almost impossible for people to have a well-informed opinion on CRISPR. Because we just spent 25 minutes getting forgive me, but a fairly base level.
No, absolutely. Like a smash and grab. I ran in and got like the 5 things that mattered and kind of threw them at you.
Right. And so, we can't necessarily look to the public for ethical considerations on things that are at a really high level of scientific knowledge, because it's not going to be an informed opinion, and seems like a case where an informed opinion is a good thing to have.
Massively important. I mean, I have a bachelor's degree in a field of science, and CRISPR is something that I barely understand.
I don't understand it at all. So
I mean, it's absolutely fascinating, absolutely mind-boggling. And so you take that and you apply it to like every single thing that you just talked about. How do you make policy for things that a select few people understand?
Yeah.
I don't know if there is an answer to that question, and perhaps this would be a great time to say, what do you think?
Yeah.
I'm looking at the camera. The answers to some of those questions might be, because we didn't really give you any.
I don't know, guys. Should we modify the human genome? Should it be Gattaca? Does Gattaca society sound good to y'all? Or?
At how much and what level of modification is okay? Because I think the concept of removing the world of genetic disease is incredible. But what's to stop us, if we do reach that point, from starting to pick traits that we want?
Right.
Where's the line?
Right.
What's okay and what's not okay? What do you think is okay and what's not okay? Tell us in the comments. There are not good answers to these questions, but we would love to hear what you have to say. As always, thank you all so much for watching, and take care, everybody.
Thanks, guys. See you next time.